Good morning. Today I, Neha Bankoti of class 10C, going to explain the study of binary fission in amoeba and budding in yeast. To start with the activity, first of all, let us understand what is binary fission. Binary fission is an asexual method of reproduction. In binary fission, the parent organism splits to form two new organisms. The parent organisms cease to exist and two new organisms come to the existence. The unicellular organism like amoeba, paramecium, leishmania, bacteria, etc. reproduce by binary fission. Amoeba reproduce by binary fission by dividing its body into two parts. When the amoeba cell has reached its maximum size of growth, then first the nucleus of amoeba lengthens and divides into two parts. After the cytoplasm of amoeba divides into two parts, one part around each nucleus. In this way, one parent amoeba divides to form two smaller amoebae called daughter amoebae. Now, let us understand this with the help of an activity. Aim to study binary fission in amoeba. Materials required are pond water in a beaker, a slide, dropper, cover slip and compound microscope. So, let's start with our procedure. Take a small drop of pond water in a slide and cover it with cover slip. Do not add any stain. Observe it under a low power of microscope. As we see, the amoeba is visualized under the low power of microscope. Observation: amoeba are usually irregular in shape. Some of them may be in state of being binary fission, in which nucleus is divided and then cytoplasm is divided, so that the amoeba divide into two daughter cells, which is known as binary fission. Now, let us understand what is budding. Budding is also an asexual method of reproduction. In budding, a small part of body of the parent organisms grow out as a bud, which then deattaches and form new organism. The organism which reproduce by budding are yeast and hydra. Yeast is tiny unicellular non-green plant which is a fungus. Yeast reproduce by budding. In yeast, first a bud appears on the outside of the cell wall. The nucleus of parent yeast cell then divide into two parts and one part of the nucleus move into the bud. Ultimately, the bud separates off from the parent yeast cell and form a new yeast cell. Now, let us understand this with the help of an activity. Aim to study budding in yeast. Materials required are yeast powder, sugar solution in a flask, dropper, slide, cover slip, cotton, and compound microscope. The procedure is: add 5 grams of yeast powder to above 250 milliliters of 10% of sugar solution in a flask. The mouth of the flask should be tightly plugged in with cotton wool. Now keep the flask in a warm place for 4 days. After 4 days, take a small drop of culture solution on a slide and cover it with cover slip. Do not add any stain. Observe it under the low power of microscope and we can visualize the yeast cell, and some are undergoing with binary fission and some are undergoing with budding stage. So yeast can go under both budding and binary fusion. Observation: yeast cells are usually spherical and oval in shape. Some may be in state of binary fission and some may be in state of. Some precautions we can take while performing both the activity. First, we should not use any stain. Second, the slide and microscope must be handled carefully. Third, focus the slide first by low power and then high power. That's all. Thank you and have a nice day ahead.